Hello, we're going to focus on this last lab with the special senses. Now, keep in mind that what we're looking at are the senses vision, olfaction, hearing, taste, and the sense of balance. Interestingly enough, all of this is pretty much involved with the head area. There's a reason for that. Your pathways of information are much shorter to the brain, which would have to analyze that special sensory information and determine if some sort of response is required. Now, you might think about that and it doesn't click, but think about it this way. Instead of my eye being in the orbit of my skull, what if it was at the tip of my finger? Hey, it's great. I can look and see what stuff is in my pocket. But that would require a much larger and much longer optic nerve to eventually go up to the thalamus and then have the information put over to the occipital lobe of the brain. So really what you have are pathways that travel back to the brain. They're short, but a lot of this is critical information, whether we're talking about hearing, the sense of balance, the sense of taste, the sense of smell, vision. So we're going to go through all of this. And yes, there is a worksheet you need to fill out and have it submitted by Friday, 5 p.m. Well, let's get started. Now, the objectives are to review the exercises in the Sarakas text. This is the lab exercise book. So it's going to be exercise 18. We are going to omit 18.4. We're going to use sense organ models and diagrams. Well, a lot of it's going to be diagrams. We may actually see some microscopic images. I will include some appropriate lab videos, which you must review because some of them are going to be doing the things that you might not be doing in the lab, in the sense that, well, they're going to probably do a nice job with an eye dissection of a cow eye. And if you're doing this course with an online component, it's a little hard to do that. But also you'll be able to have an appreciation for the various structures involved and surrounding the eye that protect the eye so that it can successfully focus and provide the visual information to the brain. Now, again, as always, your review sheets will be pages 339 to 342. They're already present in the Blackboard site. You need to download them, fill them out, complete them, and then upload them in PDF form, and they'll be graded. They must be submitted by Friday, 5 p.m. of this week. Let's get started. First area we're going to talk about is the sense of smell, olfaction. How does this work? Well, let's take a rose as we see in this diagram. And what happens is that as the fragrances come into the nasal cavity, they are picked up by the olfactory receptor cells. These bundled axons will penetrate the cribriform plate of the ethmoid bone and deliver this information to the olfactory bulb, which will process and then deliver it further down the olfactory tract to where it will be processed even more. And the olfactory information will be processed by the limbic system and the hypothalamus, as well as the olfactory cortex. A couple of pointers here. If you notice, you have the nasal conchae. The nasal conchae provide for the turbulence that's necessary. In other words, when you're just breathing, you may not always have the currents of air flowing over the olfactory epithelium, and that's where the olfactory receptors are. First off, you sniff, and doing that creates a turbulence to expose these particular odorant molecules to come in contact with the odorant receptors. Now, that's part one. Um, the olfactory epithelium is where you're going to find these extensions of the olfactory cells. It is interesting to note several things. One, these are the only cells that seem to be neuronal that regenerate. They have a lifespan of about 90 days and then die and are replaced. Two, not all olfactory receptors are the same. Think about it this way. We make on average 1.5 million new chemicals every year, and yet we have the capability to detect them. If we were totally dependent upon a receptor for gasoline, for example, the caveman wouldn't have known what it would be, and it would be sort of a wasted receptor. So you might say to yourself, well, that's strange. Well, what about the scent of roses or geraniums or, you know, um, juicy fruit gum or something else like that? What has happened is that olfactory receptors bind to different components or different parts of a molecule. If there's a benzene ring, there'll be one that will attach to the benzene ring and report it. If there is a hydroxyl or carboxyl group, they will attach to it. If there is a group that has multiple chains of ethyl groups, there might be a receptor that will attach to that. And what happens is it's more like summating all of this information and then processing it, identifying it, and maybe even connecting it to my limbic system, my memory. Sure. I remember the smell of marigolds on a warm evening in July at my house. I know what the smell of working with tomato plants is. Now, the reason I'm bringing this up is to help you understand. In this case, this is one of the few sensory systems that does what is called synthetic processing. You take pieces of the information and pull it together to make a final identification, as opposed to just saying sweet or sour. Yet, the sense of smell is absolutely essential. 80% of the pleasure that we get from the taste of food really is actually the olfactory system. I can attest to that. Years ago, I had a septoplasty, which was basically fixing the septum in my nose. And of course, after the doctor had finished his work, my nasal cavities were plugged completely with padding and cotton gauze and everything else. So I couldn't smell anything for several days. I sat down and I had a wonderful orange drink, Sumo. And you know what? I couldn't enjoy the smell of the oranges. I couldn't enjoy the smell of the drink as a whole. It was just like I was drinking sugar water because I had no olfactory sensation there. Okay, I want you to have an appreciation for that. Also, one other little detail. We have talked about COVID-19, and one of the symptoms that appears with some patients is a condition called anosmia, loss of the sense of smell. Turns out a recent paper has come out and said it's not that the virus is attacking the olfactory cells, but around the extension of these cells, where the cells come out, they have supportive cells, and it is the target for the COVID-19 virus. When those sensory cells are damaged, then the sense of smell is declined. But after the person recovers from the COVID-19 infection, the sustaining cells will eventually recover, and then the sense of smell will recover as well. So let's look at this. We're going to talk a few minutes about the microscopic anatomy. And at the bottom, you can see where I've already answered that question. What is anosmia? The loss of the sense of smell. Olfactory receptor cells, these are bipolar neurons that are stimulated by chemical substances, odorants in the nasal cavity. The supporting cells, these are columnar epithelial cells that surround the olfactory receptors. Basal cells divide regularly to produce new olfactory receptor cells. Olfactory glands, these are the glands that secrete mucus onto the surface of the olfactory epithelium. Got to keep the olfactory epithelium moist. Cilia are located on the olfactory receptor cell surface, and the receptors are actually located on the cilia. Also, now there's an activity of performing an olfactory adaptation test and a result. You may not have the availability of that. You could take anything that you find very fragrant, whether it's a rose or something else like that, and do the experiment yourself. There are several things to be mindful of. Olfactory adaptation is much slower as we get older. It is much, the better way to put it is, we tend to detect smells. Let me reiterate. Olfactory adaptation, which is getting used to the sense of smell, occurs in a much quicker time for those who are young or adults. It takes them much longer for those who are elderly. All right. And the adaptation is the decreasing sensitivity to a stimulus over time. The sensitivity to an odor diminishes during continuous stimulation. For example, wearing perfume. You're exposed to continuous stimulation. That's why you may notice some people who are older who don't detect the scent of the stimulation as easily, excuse me, and they may actually think that they haven't added the perfume. And then you have someone who is very young, whose nose is very, very acute in detection of that perfume. They'll be sitting there saying, well, why is grandma smelling so much like her perfume? And really what it is that grandma's olfaction and her adaptation is much diminished. And so she thinks putting on more will finally give her the sense of detection, and it will be there for a while. Unfortunately, for the rest of us, it gets overpowering. Hope that helps. Now, here's what I was talking about earlier when I talked about COVID. If you take a look here, here's an olfactory gland releasing mucus, and the olfactory dendrites, these cilia here, they contain the receptor proteins. There's a knob structure here from which many of the dendrites kind of erupt from. Here you have a mucus layer. Then you have a supportive cell, the olfactory receptor cell here, and then of course it has these extensions, these axons that go up through the cribriform plate up to the olfactory bulb. Now, this area here would be referred to as the olfactory epithelium. This would be the supportive cell that assists for the functioning of the olfactory cell. All right. Now, the basal cells are right up close against the lamina propria. They would undergo mitosis, divide, and replace dying olfactory cells that are worn out usually. One other thing. You might ask yourself, well, okay, so olfactory receptors are in this sort of mucousy, watery environment. So does that mean that something that does not give off vapors or is not soluble in some sort of liquid environment such as you see here would not be necessarily detected as a smell? That is correct. Okay. By the way, if you take a look at this image, you have a developing olfactory receptor cell here. Eventually it will come out, sprout out, and then basically set up its own network of dendrites. Now, when we examine the anatomy of the gustatory structures, we identify the following structures of the oral cavity: the tongue, the pharynx, and the epiglottis. Now, sensory receptors for taste are located on the taste buds. These exist on the tongue as well as the soft palate, pharynx, and epiglottis. The papillae of the tongue, there's three primary types. Circumvallate contains more than usually 100 taste buds. Fungiform contains 5 to 10 taste buds. Filiform lacks taste buds completely. And you can see here's the tongue here. Here would be the pharynx, okay? And if we look in the tongue itself, we see the valate, also called circumvallate, the folate, the fungiform, and the filiform papillae. What you see also, and you can see the valate papillae here, all through it here, we have water receptors in the back near the pharynx. These also have detectors for an unusual sense of taste. We're all familiar with sour, bitter, salty, sweet. But there's another one called umami. Now, umami is a Japanese word for deliciousness. And what happened was, in about 1962, Japanese scientists were exploring this sense of taste that involved things like meaty, brothy taste. Okay, they were able to finally isolate the umami receptor, which reacts to things like the glutamate that's present. And so you really have 5 senses: sour, bitter, salty, sweet, umami. Now, if you notice, the valate has their taste buds up and down on these rows here. The taste buds would be here for folate, here, and here for the fungiform. You don't really have a lot here with the filiform yet. But when we examine this, the taste bud microanatomy, the gustatory cells have a lifespan of about 10 days. Basal cells divide to produce transitional cells, and the transitional cells transition into gustatory cells. So basal cells begin the process to provide replacements for the now functioning gustatory cells, but those that fade out, they'll be replaced. The types of taste I've mentioned. You want to skip the gustatory test, which is activity 18.4. Here, we're going to see the microanatomy of a taste bud. If you take a look here, here are our taste buds here, here, here. And if you get up close, you're going to see the following, just like this diagram, which allows it a little bit easier. You will have a transitional cell, a gustatory receptor cell, which on the end of it has taste hairs. These are microvilli with receptors that will respond to the various chemicals, whether it's sweet or sour or bitter, etc. But each of them are then connected at the back end to axons of the sensory neurons. The basal cells here are ready to divide and replace the transitional cells, as the transitional cells are eventually act as a replacement for these receptor cells that basically will eventually die out. They only last 10 days. And so you can see how all of this is wrapped around the taste pore. So all of the taste hairs focus out into the taste pore. All right. And you can see how these caverns and craters are. Basically, you have the taste pores facing outward here, etc. here, so they can come in contact with whatever we are consuming. The next area I'm going to move into is looking at the anatomy of the accessory eye structures. Now, the outer structures are somewhat protective in one way or another for the eyes. Eyebrows are thick hair bands. They protect eye from perspiration and direct sunlight. Eyelashes are thick eyelid hairs, and they protect eyes from particulate matter, dust and small particles and things like that. The eyelids, the palpebrae, are fleshy eye coverings. Blinking the eyelids moistens the eyes. They contain modified sebaceous glands, meibomian glands. These secretions prevent the eyelids from actually sticking together. Now, the conjunctiva lines the inner surface of the eyelid, and you're going to find that there are 2 components of the conjunctiva. There's the palpebral conjunctiva, which is on the inner surface of the eyelid, and then the ocular conjunctiva, which is on the anterior surface of the eye. We also have the palpebral fissure, and then we have the medial canthus. You'll also find out that there is a structure called the lacrimal caruncle. It's a reddish mass at the medial canthus, and these glands produce mucus while sleeping. So here is the lacrimal caruncle. Here is the medial angle of the eye, and of course, this is the lateral angle of the eye. Here are the eyelashes. Eyebrows would be up here. Here we have the cornea, which is the clear area. Note there are no blood vessels in here. How the cells for the cornea get the nutrients is by diffusion from the fluid that is inside here. We'll get into that in a minute. The aperture that allows light to come onto the inside of the eye is called the pupil, and obviously what controls that in part is the iris here. We'll get more into that in a minute. The palpebral fissure is right here. Here's our eyelid. Okay. Now, if you move to the side, we're going to be talking about the lateral canthus and the lacrimal apparatus, which is a very interesting system. The lacrimal gland is located inside of the superior-lateral margin of the orbital cavity. It's a mouthful, but what you want to keep in mind is that lacrimal glands are absolutely important. They provide the moisturing of the eye surface. The lacrimal ducts transport tears to the surface of the eye. The lacrimal puncta is a drainage pore that is located on the lacrimal caruncle. It is the lacrimal sac that you get the drainage from the lacrimal puncta, and then from there, the nasal lacrimal duct drains the tears from the lacrimal sac. Now, tears contain an enzyme called lysozyme, which kills most bacteria. Okay? To help you to understand this, think about it this way. All right? There is the lacrimal gland here. Here is the tear ducts. They eventually will release tears into the eye here. Eventually, what happens is you have the lacrimal puncta right here, all right? And this carries the liquid, the tears, to the lacrimal caniculi, which eventually empty into a lacrimal sac. That empties eventually into a nasolacrimal duct, and you have it drain into just below the inferior meatus. Now you might say to yourself, okay, but why is it so important? Well, think about it this way. You ever notice if you cry, you eventually have to blow your nose? Well, the reason that you do that in part is because some of the tear material will actually get into the nasal cavity through this passage system. Next, keep in mind that the eye in the orbit also is supported and protected, some of it by orbital fat pads, but the support also comes from the various muscles. We have the superior rectus muscle, we have the inferior rectus muscle, and the inferior oblique muscle. We're going to get into the muscles in a second, but think about how you're able to rotate your eye all in all the different directions. Here is our, of course, upper eyelid, the lower eyelid. Now, let's talk about the gross anatomy of the eye as it's sitting there in the orbit, which that bony orbit, you have also cushioning by a layer of fat. The eye is supplied by optic nerve number 2, CN2. Okay, that's fine. That's the optic nerve. You want to locate the tunics, or the layers of the eye. You have 3 of them: fibrous, vascular, and also the neural tunic. The fibrous tunic is the one that we see. The sclera is the tough, opaque membrane. It contains collagen and elastic fibers, protects the shape of the eyeball, okay, and pretty much is opaque, so light will not come in except through the cornea. The cornea, on the other hand, is transparent to allow light into the eye. It lacks blood vessels, whereas the sclera has blood vessels. The vascular tunic is referred to as the choroid. It contains numerous blood vessels and melanocytes to produce melanin, which block light reflection out of the eyes. The anterior extensions include the iris, which controls the light entry into the eye, the ciliary body, which is the ring of smooth muscle that adjusts the shape of the lens for focusing. Now, the lens is a transparent structure. It adjusts the focus of objects against the retina. One thing to keep in mind, the lens. If you've ever heard of someone having cataract, what happens is you have these very special crystalline proteins that are basically transparent. Due to age and overexposure of either ultraviolet light or infrared light has been found to cause a yellowing, a congealing of some of the proteins in the lens, and eventually over age, you get a point where you may have to get an artificial lens because you've got the development of cataracts. In other words, opacities that develop in that lens. Today's modern medicine allows us to put an artificial lens in there, excuse me, and it is amazing what people report after they've had this transplant put in. All right. Now, the neural tunic, this is where we get into really the important area. This is the retina. It contains rods. Now, rods are effective in the detection of light, either extremely low level light or great amount of light. Cones detect color. So if you get confused at times, cones color, rods light. The cones are set up on a triad system: red, blue, green. In those three colors, you can have variations in mixtures that will allow you to account for colors like, salmon, orange, purple, yellow, etc. If you can see from this diagram here, a couple of things to keep you in mind. Here is the corneoscleral junction, right here. Here we have the beginning of the cornea. Down over here, all the sclera. Underneath the sclera is the choroid, the rich blood vessel system throughout. And underneath there would be the inner layer or the retina. A couple of things also to note. You have a structure called the iris, which basically creates the pupil, which controls the amount of light to come in. This inner lens here plays a key role in focusing of the image against the back of the retina. But how does it adjust? Well, that's via the ciliary body with basically these ciliary fibers here that are attached to the sides of the lens. Excuse me. Now, as these, if you want to have very fine detail, you might have this contract. If you want to have distance viewing, you would relax the lens. One of the things that happens sometimes is that as we get older, the location of these fibers drifts as the lens gets older. And that's when you start having people over 40 having to carry reading glasses again, because their focus is not close enough to their face as it was when they were 20 or 10. And now they have to have adjustments to that. That's called presbyopia. Excuse me. So the retina contains rods and cones, and there are three different types of cones. As I said, one's red, one's green, one is susceptible to blue. We have a specialized site on the posterior surface of the retina. several of these structures. We got to keep mindful. The Macular Lutea is an oval yellowish area surrounding the fovea near the center of the retina in the eye, which is the region of the keenest vision. This particular site, when we talk about the fovea centralis, is loaded with cones. That's where you get the sharpest visual image. Okay, so that's the central section of the macula lutea. There's one thing to keep in mind. If it's all cones, which means you get fine detail of color and everything else, there's no rods. If there's no rods, then you don't have the capability to see in low light. We'll answer that in a minute. The optic disc. Now, we have to think about this. All of these retinal cells have these fibers, axons, basically coming together into one spot and forming the optic nerve to go beyond the eye, outside the eye, okay? So that's where the optic nerve exits. And so as a result, on that particular spot, we call that the blind spot. You've ever seen those exercises, you might even see it in the book, where you look intently with one eye and you have a little box. And it's got an X and it's got a circle. And if you look at the X and you keep bringing the image closer and closer, you will find a point where the circle appears to disappear. And that's because the circle image came over the area of the optic disc. So it appears to be magic, disappeared. It's not magic. It's just the image went over that site, so there's no way to detect it. Now, here's another little interesting point. So we talked about the optic disc right here. You notice all the blood vessels that come in and out? That's all part of the optic nerve. Blood vessels are in the center, they come out through, and so they feed the eyeball. You have a central retinal artery and vein, they emerge from the center of the optic disc. You have the macula, and you have the fovea centralis. This is the dense area at the center of the lighter macula. What happens when we have to adjust to light and dark? If you were a Moody Blues fan of that rock band many years ago, they had a song called Nights in White Satin, and at the end he starts talking about, almost in a poem, talking about the moon, strange, moon and strange colored orb of the night. And he starts talking about how yellow becomes white and red becomes gray, etc. What he's really talking about is the transition going into night vision. What I mean by night vision is this. At a much lower amount of light, you're not going to see things. Not at the fovea centralis, because you need a much greater amount of light for the activation of red and green and blue. So if you have difficulty looking exactly on something and trying to read it or see it with very low level light, the trick of it is look at it off center, as if you were looking at it, which it's located, let's say, around that dotted line, but you're looking with your view being somewhat over near the optic disc. You'll see it a little bit better. What happens is you want to move the image to where there's areas of more rods, which are sensitive to low light. Okay. You probably have also known the phenomenon which occurs when you go from a brightly lit room to a dark outside, and it takes time for your eyes to adjust to the differences in the light intensity. You will see a lot less in the sense of color. It'll become more whites and blacks and grays and things like that. Okay, I hope that helps. Now, here's another weird, surprising thing. Here's my optic nerve. Here's my central retinal artery and vein. Here's my sclera. Here is the inner part of my eye. Here's the blood vessels coming out. So this would be my optic disc. And blood vessels supply the inner layers of the cells. Here's my retina, but here's the thing. Right here are some pigmented cells, sort of act as a backstop so light doesn't bounce back in the eye. Kind of stops it. And yet the retinas are pointed in the opposite direction from the light. Whether they're rods or cones, they're all in back here. They are then connected to another layer of cells. These cells are called the ganglion cells. And they're connected to another layer of cells here. And there's also two layers of intermediate connecting cells here and another one here. And eventually these ganglion cells right here, sorry, these were the lower cells. These are, I've got to find that, but I think it's basically, these are some of the photoprocessing cells. These are the ganglion cells because they're going to connect and they're going to have very long axons that are going to head down this way and be part of the optic nerve. Why am I saying this? Well, if light comes down through here, it's got to go through these layers of very clear cells, but eventually it has to come down into here to activate, stimulate, and fire off these cells to be receptive of an image. Okay? By the way, you have to also consider something else here, and that is the chambers that are in the eye. Now, the anterior chamber is filled with a watery medium. It's called the aqueous humor. Of that, there's two chambers, an anterior chamber between the cornea and the iris, and a posterior chamber between the iris and the lens. Now, the posterior cavity is filled with a gel-like vitreous humor and helps to maintain intraocular pressure. When you see the video on a cow eye dissection, you will notice that as they begin to open up the eye, there'll be this jelly-like mass that comes out. That's the vitreous humor, okay? There's one other thing that's interesting. If you look with certain animals, not humans, but it's found with many nocturnal vertebrates, raccoons, skunks, opossums, rabbits, also domestic cats and dogs. You may have had this where it's dark out and you flash a flashlight and you see their eyes sort of like glow back. Well, what it is that there's a layer, a reflective layer just behind the retina called the tapetum lucidum. And what that does is enhance low light vision for these animals. They can see far better at low light because what this does is it acts as a reflector and reflects back photons back again to the retina to provide for more visual stimulus and information. So here is my posterior cavity, and this is my anterior cavity. This is filled with a jelly-like material, clear. It's called the vitreous body. Here's my lens. Here are my ciliary filaments, the ciliary body, and then the iris. Now here, I am secreting a fluid in this anterior chamber. This fluid will travel out through and then out to the back over here. This fluid will also provide nutrients and gases, etc., for some of the cornea cells, etc. This is the fluid that we try to monitor so that we don't have too much pressure, which can lead to the beginning of glaucoma. Because what happens is as the pressure builds up in the eye, it starts to act on interfering with the circulation of the blood vessels in the back part of the eye, leading to the death of the retina, and it would eventually deteriorate going backwards. That's where they get that tunneling vision effect. Now there is a small sinus right here at the junction between the cornea and the sclera that absorbs the excess liquid. And there is some tissue here at the ciliary body that produces this liquid, okay? Now, I forgot those words, but I now brought them back. Bipolar cells, ganglion cells, etc. When we have the retina, the light enters from the outermost layer and strikes the cones and the rods, which are in the deeper layer. Now, we're approaching from the outside, we're approaching from the back. So we come through the ganglion layer, which are axons of these cells make up the optic nerve. The next layer is called the bipolar cells. These cells synapse with rods and cones. And then we get to the photoreceptive layer, which is where you have the rods, which detect light of various intensities, and cones, which give you sharp vision as well as color vision. Now, this light-sensitive section is actually pointed away from the light direction. We have a pigmented layer, which is produced by melanocytes that are producing melanin, and they prevent the light being reflected. Now, you might be able to perform visual tests for binocular vision and depth on the pages 322 and 323, as well as the optic disc exercise, which is on page 321. If you don't have the time or you don't have the interest, don't do it. Don't worry about it. But they do provide other means to test this out. It's easier to be done in a lab with the necessary equipment. Unfortunately, if we don't have that capability this semester, don't worry about it. Just be familiarized with things like the eye chart, etc. Here's where light's coming in. It passes through this layer of the ganglion cells, it passes through this layer of the bipolar cells, and then it's going to activate. And notice that the stack of coin-like structures for the rods, as well as the coin-like structures for the cones, they're all small here. These actual points right here, almost intimately up against the pigmented layer, that's the real component of the cell that acts or reacts and detects light. Now, why do I get so excited about this? Think about this. All of this is in the opposite direction of where the light is coming through. Then you have these unusual cells that kind of join a bunch of them together. And it's called horizontal cells. They may process and exchange information, etc. Then you've got this other group that attaches between bipolar and the ganglion group, and they're called amacrine cells. They also will exchange information, modify the signal, etc. Now, you might sit there and say, isn't that a lot of minutia? No, because for one thing, damage to amacrine and horizontal cells will tend to reduce the visual accuracy. But another way to think about it is this. Human vision is such now that in a completely dark environment, you can tell if somebody strikes a match in the dark, well over a mile away, and you can see that flickering little flame. That's an incredible capability of visual acuity and visual processing, wouldn't you say? Now, there are a few other tests you can do: the near point accommodation, the visual acuity, the visual tests for astigmatism, pupillary reflex, color blindness. I'm not going to put emphasis on them now. If I can, I'll try to get a little bit of video on them to demonstrate them to you. What you want to keep in mind with the pupillary reflex is that in part is absolutely essential so that we are not overexposed to high intensity light. And this is part of some of the various reflexes that occur. With astigmatism, we have other defects along the surface of the eye that can cause problems with visual focus. Okay? And with color blindness, a lot of that is a genetic characteristic. A lot of times it's been found on the X chromosome. There are three major types of color blindness. When I say by color blindness, it's not that you don't see the color. You can't discriminate between two colors. So let me give you an example here. Red, blue, red, green. Red, green is the most widely known, and blue, green. Blue, green is the rarest of the forms of color blindness. And there seems to be slight defects in some of the genes that are involved in some of the proteins and some of the products that are involved in vision in those cells, so that individuals cannot tell clearly if something is, let's say, blue or green, or they would look at a large array of colors and see a much greater reduced amount. And this can cause problems if you're just working on things like wiring that's based on colors, or if you're looking at stop signs. Fortunately, we have stop signs. You have the stoplight, one in the top, one in the middle, one in the bottom. And as one person said to me, no problem, I don't worry about it. They all look green to me. I know the one on the top is red. I know, and that's stop. The one in the middle is warning, yellow, and the one on the bottom is go. Okay. Here's an example of the pupillary reflexes. We have two sets of muscles in here that are smooth, the dilator pupillae here and the sphincter pupillae here. Decrease light intensity, increase sympathetic stimulation. What happens is that you end up with a dilation of the pupillary, and so that this contracts, and so you have more light being able to come in. If you have an increased light intensity or increased parasympathetic stimulation, this sphincter pupillae will contract, and you will have a much smaller pupil here. All right, we move from here in the eye to the ear. We're going to go over some of the basic structures first. The external ear, the auricle is called, also known as the pinna. It is a helical structure with a cartilaginous rim. The lobule or earlobe is the fleshy part. The external acutus meatus meets with it, and you have also the tympanic membrane, otherwise known as the eardrum. In the middle ear, this is an air-filled chamber lined with mucous membrane. You have present in it the auditory ossicles, which, if you remember, we had dealt with those. They are bone structures. You have the malleus, which is known as the hammer, the incus, which is known as the anvil, and the stapes, which is known as the stirrup. The malleus is attached to the tympanic membrane. So as the tympanic membrane moves back and forth, the malleus will move back and forth. The malleus will transfer vibrations to the incus, the anvil, which will then transfer the vibrations to the stapes. The stapes is attached to the oval window of the inner ear, the cochlea. These vibrations will create vibrations in the fluid environment of the inner ear, to which we'll get into in a few minutes. The pharyngotympanic tube, otherwise known as auditory tube, otherwise known as eustachian tube, allows equalizing of air pressure from the nasopharynx to the middle ear. If you've ever been on an airline and you have ever had it where you start feeling this pressure and it's like suddenly you're not hearing as clearly. Airplanes have to go up to 25 to 33,000 feet. Okay. But you have a pressurized cabin. But the pressurized cabin is not perfect at 5,000 feet or even at sea level. They really bring it up to about, on the average, the pressure in the cabin is about the equivalent of 8,000 feet. It's still higher, but it causes some pressure distribution for some individuals. Now, if you've ever been, unfortunately, on a plane with a child crying, it's because they're sitting there going, it hurts me, but they can't articulate that. That's why mom gives them a drink or something else like that in a bottle, and hopefully they're healthy, they're happy. Actually, the drinking, the sucking motion will keep opening up the eustachian tube sufficient to cause the re-equalization of the pressure. With a lot of us, what we'd have to do is kind of block your nose, breathe outward with your mouth closed. That will cause the pressure change that will pop open the eustachian tube and equalize the pressure between the middle ear and the outside environment. Here is the auricle, the elastic cartilage, the external acutus meatus, the tympanic membrane, Here's our tympanic cavity, auditory ossicles, hammer, anvil, stapes, and these are attached to this very long structure which is embedded in a bony labyrinth, and this is the cochlea. Now the cochlea processes sound information. There is another set of semicircular canals that will process balance. You have a vestibular cochlear nerve here, and part of this is because you have the cochlear nerve, which fuses with the vestibular cochlear, and of course, then you have the vestibular nerve that fuses along with it, creating cranial nerve number 8. Okay, we're going to get more into this in a minute. And here, by the way, don't forget, is the auditory tube, which then goes to the nasal pharynx. Okay. This is much more up close on the auditory ossicles, the malleus, the incus, and the stapes. There's 2 things to bring up also. There are 2 very small muscles in the inner ear. The tensor tympani muscle right here. Notice that it has a grip, if we'll say, on the hammer. We also have another one called the stapedius muscle, which has an attachment to the anvil. What these will do if a buildup of high sound occurs is by a reflex is begin to pull away some of these and reduce the intensity of the vibrations being fed into and through the ossicles to the inner ear. Now, unfortunately, it doesn't work when you have such rapid changes in sound, such as a backfire of a car or a shotgun or some things, but it allows for stabilization and protection of very loud sounds. You'll also notice that there's a branch of the facial nerve, number 7, which is kind of cut here. And you'll notice the tympanic cavity here. This is an area that is very prone to having some bacterial infections go in, leaving otitis media. And so you either try antibiotics, and if the bacteria are responsive, great. If not, the doctors will sometimes do a tympanectomy with putting in a little tube in here to allow the leakage of the bacteria and pus, etc., out. And because this is sort of an area that gets semi-anaerobic, organisms will be here, but when the air comes in here, it alleviates and reduces the infection. As years have gone on and as more microorganisms have become resistant to antibiotics, putting tubes in ears has become much more the useful strategy. It was big in the 50s, and then we had the antibiotic period, and then we started having the rise of antibiotic resistance, and so it's now being turned back to an effective strategy to deal with inner ear or middle ear infections. So, as I mentioned before, the 2 middle ear muscles protect the hearing, stapedius and tensor tympani. The inner ear is called also the labyrinth. In that structure, we have the vestibule, which is consisting of the utricle and saccule, and the macula. The macula are sensory receptors. They detect linear movements and respond to gravity. The 3 semicircular canals are semicircular tubes that are connected to the posterior wall of the vestibule. One end of the canal has a structure called an ampulla, which is an enlarged region with crista ampullaris. These are sensory receptors that detect changes in the angular acceleration or deceleration. We'll show you that in a minute. It's sort of like it has a gel in it, and it pulls on certain sensors. Now, the cochlea is a snail-shaped structure, and what it is basically it's a spiral type structure, but it looks like one of those big spiral snail shells, even though it is very, very small. And it contains sensory receptors for hearing, and this is called the organ of Corti. Here is the structure. First, we'll deal with the membranous labyrinth. Now, what you have to understand is there's fluid in here, and the fluid is either perilymph or endolymph, and they can't be mixed up, and I'll tell you why. because perilymph has a different ion composition. I believe it has to do with sodium and several other ions. Endolymph has a different type of makeup. If the two are mixed, the tissue begins to die. Now, what seals these semicircular canals is that they're wrapped in bone, and then you have this fluid, and then you have the membranous labyrinth, and then you have endolymph in here. If you note, the semicircular canals are set up somewhat like the X, the Y, and the Z in spatial orientation. Okay, so basically, one is forward, backward, one is up and down, and one is side to side. And you have both of these. You have one set on one ear, one set on the other. All right. And what happens here in places like the vestibule, which is right in here, is that you're going to have sensors that are going to detect movement of fluid. And the movement of fluid is going to occur because when you move your head side to side, up and down, jump up and down, etc., you're having flows that change and they will pull on these receptor areas. Now, besides the semicircular ducts, you have the utricle, which is right in here, and the saccula, which is right down here. All right. Now, we haven't got into the cochlea yet. We'll get into that in a little bit. But it's important to understand what is membranous versus what is bony. Now, let's examine first the anatomy of the cochlea. Bony snail-shaped structure consisting of 3 chambers. In these chambers, you have the vestibular duct, which is part of the bony labyrinth, and it's filled with perilymph. Begins at the oval window and ends at the apex of the cochlea. The cochlear duct, part of the membranous labyrinth, middle chamber filled with endolymph, spiral organ located in this duct. Tympanic duct, part of the bony labyrinth filled with perilymph, begins at the apex of the cochlea, ends at the round window. Now, if I was to take this coiled little structure and stretch it all the way out, what I'd have is one chamber, okay, that was continuous from the round window, one chamber that was continuous from the oval window, and in between each of these two chambers is this organ of Corti. Now, the organ of Corti feeds nerves back through to the cochlear nerve, which then again fuses with the vestibular nerve, okay? And what you have are these spiral ganglions, the scala tympani, the spiral organ, the vestibular duct, and the scalar vestibule, right? Okay, now, as we move forward here, we've got to recognize some of these other structures here. This is all very important in hearing. When we talk about the vestibular membrane, it separates the cochlear duct from the vestibular duct. The hair cells are located in spiral organ and contain stereocilia for sound frequency detection. The spiral ganglion, the neuron cell bodies of the cochlear nerve this is all part of cranial nerve number 8. The tectoral membrane arches over hair cells, and the hair cell stereocilia are embedded into this membrane. The basilar membrane separates the cochlear duct from the tympanic duct. Now, if it helps you as you're doing this, have your text, Syracuse, opened up to see where these structures are, okay? The Weber-Rhine test you really need the equipment for, but I have video that is included in there to show you it, and the hearing acuity and sound localization test as well. Here's the main point. Here's the scalar vestibule. Here is the cochlear duct, okay? Here is the scalar tympani. What you're looking for are these little structures in the cochlear duct. This is where the organ of Corti is. If this section vibrates, it's going to pull on the membranes and it's going to affect the organ of Corti. All right, I'm not done here yet, but what I want to help you to understand is this. There is the tectoral membrane here. There are cells. They have hairs. They're embedded in the tectoral membrane. And as the entire structure moves, they will pull on the hairs. These hair cells, as they're called, will fire and then pass the information over to the cochlear nerve. To help you understand it, if I took this big snail structure and just rolled it out a length, and you'll go and understand this better when we go through the chapter 15. Here's the end result. Certain parts along the length react to high frequencies, others react to low frequencies. So as I'm talking to you, or the next song that you like, you're listening to, you will get a variation of parts along this entire organ, along the entire cochlea that will be active, bouncing around, etc., pulling on the various organs of Corti, signaling what the sounds are. Now, to help you also, one other thing. Many of you have seen how it's been a remarkable development of technology to make an artificial cochlea, or a cochlear implant, it's called. Literally what they do with microelectronics is they are able to send a very fine microelectrode through all of these areas here. And at the same time, the microelectronic wire has areas that would fire the sensory and activate it such that the computer that assists this breaks down the sounds of the various frequencies, and the individual is starting to actually hear the various sounds, high, low, medium frequencies, and its intensity also. And so they are suddenly amazed that they can hear things that they did not hear before. I hope that helps you. It's just to get you a beginning of this. At this point, I would encourage you to review the exercise 18. Remember to omit 18.4. Use the nerve and the spinal cord models and reflex diagrams. It's not going to give you as much, but I would say to you instead, review the microscopic slides and the images I've included in the PowerPoint. Review the videos that I have included, so you can review the sense organ models and some of the applications. You're going to have to use Cerecus as well, your exercise book. Take the exercise sheets on pages 339 to 342. Complete them. Make sure that when you upload everything, that it's uploaded in PDF. Now, for some of you that may have some glitch where you start writing on one line and then something happens and it repeats what you just wrote on the second line, one of the suggestions I gave to a student was write the first line, save it, then write the second line. You shouldn't have that replication occurring. The main point is fill out all your work so that I can give you the grade that you truly deserve. Remember that you have to submit this uploaded at the Blackboard site prior to 5 p.m. on Friday. Take time to review the lab videos. Now, obviously, we are done with all our lab exercises. So the next step is going to be lab practical exam number 2. I would encourage you to prepare for that. Go over all of the handouts, the videos, the sections of the text, including micrographs and the diagrams, etc. Okay? And I hope you all have a very nice day.